Okay, this section is the last section of looking at Unit 3, Section 8, which is amino acids and proteins and DNA, and we're going to look at DNA. So DNA, it is a polymer of nucleotides. So DNA, as you probably realise, stands for deoxyribonucleic acid, DNA. And DNA contains all the genetic information of an organism. And DNA is made up of lots of monomers, and these monomers are called nucleotides. And the nucleotides are made up of the following three things. They're made up of a phosphate group, a pentose sugar, and a base. So we're just going to have a look at those now. So we're going to look at the general drawing. So you'll often see DNA drawn out, well, a nucleotide of DNA drawn out like this. This looks like its general drawing for it. And this would be our phosphate group, this is our sugar, and then this would be our base. So let's look at this phosphate group in greater detail. What is this phosphate group? What's it made up of? And what does it look like? So the phosphate group would look like this. It is important that you know the structure of the phosphate group and that it has this slight polarity, this slight negative charge here. And this oxygen, we've got that double bond oxygen there, and then phosphate. So that is our phosphate group, which is here on our nucleotide. This is our phosphate group. So that is what your phosphate group is. You also have a pentose sugar. So I'll show you the pentose sugar now. Pentose sugar looks like this. Obviously, it's in a pentagon shape. So we've got our oxygen. So pentose sugar. A pentose sugar is a 5-carbon sugar. And in DNA, the sugar is specifically 2-deoxyribose. You don't need to know the structure of phosphate and the 2-deoxyribose or of the bases, as they'll be in the data booklet that you're given. But it's just good to know that this is what it looks like, just to sort of familiarize yourself. And then finally, we have a nitrogenous base. So each nucleotide is made up of one of the 4 nitrogenous bases. And the nitrogenous bases consist of adenine, guanine, cytosine, and thymine. So if we have a quick look at them, if I draw out adenine for you, got our NH2 at the top. And again, this will be given to you in that data booklet. So I'm not going to spend too long going through all of them. But what's really important is this is the molecule here. This nitrogen here is what will bond with the deoxyribose. So bond with the sugar. So that nitrogen there is what would bind. And so that one is adenine. And then guanine, I'm just going to draw for you now. Again, that's the nitrogen that will bond. And then I'm just going to draw you, so that is guanine, so that's adenine, that one's guanine, and now I'm just going to draw you cytosine and thymine. Cytosine and thymine, slightly smaller molecules. Again, it bonds with that nitrogen here. And this one's cytosine, and then thymine. Bonds with that nitrogen. And that one again is thymine. So then you can have all the structures of the 4 DNA molecules. So basically one of them would then join onto a ribose and a phosphate group. It's given to you in a data booklet. And what we'll do at the end of this session is I'll do a couple of exam questions where you actually get asked to draw them all together, and we'll look at how they would look like. So basically from the structures, you should be able to work out basically the structure of a adenine nucleotide, for example, from the information that's given in the exam data booklet, if you need to. So you don't need to learn them, but you need to know where that phosphate group and the base is joined to the sugar. So I'm going to show you one example, and then we'll go through some more later. So if you were to draw an adenine nucleotide, you would look for the phosphate group in the data booklet, and you would draw down your phosphate group straight away. And your phosphate group always joins at where that OH would be at the bottom. Okay, so it bonds there. And then it's going to bond to your pentose sugar. I'm not going to draw on all the parts of pentose sugar. This much is fine. And then obviously it joins at that nitrogen, your adenine. So if you were then asked to draw guanine, it would just be then this part of the molecule that you would change. So nucleotides join together to form a polynucleotide chain. So then you could have one adenine nucleotide and it could join to another adenine nucleotide. And they do this to form a polynucleotide chain. So if we drew it as that basic structure like we had earlier, your phosphate groups here, oops, your sugars there, and then your bases. This is roughly what it would look like. I know all your different bases, so it might be A and T there, for example. So they join together to form a polynucleotide chain, and covalent bonds form between the phosphate group of one nucleotide and the sugar of the other. So we've got these covalent bonds joining here. And this makes what's called, this whole area here, is the sugar phosphate backbone. So DNA is formed by condensation polymerization, and the sugar phosphate backbone of DNA is formed by condensation polymerization. So what happens is we've got a nucleotide here, or whatever base it is, and we have this OH group here. And what happens is it bonds with the OH group on the other, and we lose that molecule of water. So we lose that hydrogen here and that OH group there. So we lose that molecule of water, and we get that bonding happening between that oxygen here of the sugar to the phosphate group. So what we get forming there is when that molecule of water is lost, condensation polymerization happens, and then the bond that's formed is a phosphodiester bond, and it's covalent. So this is a covalent bond that is specifically called a phosphodiester bond. What you will notice is there's still OH groups at the top and at the bottom of the chain, and that's so further links can be made. And this allows the nucleotides to form a polymer made up of an alternating sugar phosphate chain. You can obviously repeat it by going through hydrolysis and adding the water molecule in to break the nucleotides apart. DNA forms a double helix. It's made up of 2 polynucleotide strands. And the 2 strands spiral together to form a double helix structure. And that's held together by hydrogen bonds. Each base can only join with one particular partner. So, quick reminder, adenine always pairs with thymine, and guanine always pairs with cytosine. And this causes the 2 strands of DNA to be what we call complementary. And this means that they match up so that whenever there is an adenine base on one strand, there will be a thymine base on the other. And wherever there is a guanine base on one strand, there will be a cytosine base on the other. So hydrogen bonding causes the bases to form specific pairs. So as we just spoke about, the complementary base pairing exists in the DNA helix, where adenine always pairs with thymine and cytosine always pairs with guanine. It happens because of the arrangement and number of atoms in the base molecules that are capable of forming hydrogen bonds. So a hydrogen bond can form between a polar positive hydrogen atom. So when we find a polar positive hydrogen atom, it's usually a hydrogen that's attached to anything highly electronegative, like nitrogen, and a lone pair of electrons on a nearby oxygen or nitrogen atom. To bond, the 2 atoms have to be the right distance apart. So let's look at adenine and thymine. So adenine and thymine in this example have the right atoms in the right places to each form 2 hydrogen bonds. So we've got that positive hydrogen here bonding with that lone pair of electrons on that nitrogen atom over here. And then we've got the positive hydrogen here bonding with the lone pair of electrons on the oxygen over there. So they can pair up, and we have 2 hydrogen bonds forming between adenine and thymine. If we look at guanine and cytosine, we've got a positive hydrogen here and a lone pair of electrons on the cytosine, so we get 1 hydrogen bond. We've got another positive hydrogen on the guanine and a lone pair of electrons on the nitrogen on the cytosine, so we get a 2nd hydrogen bond. And then finally, we've got a lone pair of electrons on our oxygen on the guanine and a positive hydrogen on the cytosine, so we get 3 hydrogen bonds forming between cytosine and guanine. So they can pair up too. These are the only possible base combinations. Other base pairings would put the partially charged atoms too close together, so then they would repel. Or they'd be too far apart, or the bonding atoms just wouldn't line up properly. The DNA helix has to twist so that the bases are in the right alignment and at the right distance apart from each for the complementary base pairs to form. And finally, we're just going to look at something called cisplatin. And cisplatin can bind to DNA in cancer cells. So cisplatin is a complex of platinum 2 with 2 chloride ion ligands and 2 ammonia ligands in a square planar shape, which you can see here. And it is used as an anti-cancer drug. And as you can see on this image, the 2 chloride ions are next to each other. So this complex is cisplatin. If they were opposite each other, you would have transplatin, which have different biological effects. And as you know, cancer is caused by cells in the body dividing uncontrollably. And as they divide uncontrollably, they form tumours. In order for a cell to divide, it has to replicate its DNA. And what cisplatin does is it binds to DNA and it causes kinks in the DNA helix. And when these kinks form in the DNA helix, it stops the proteins that replicate the DNA from copying it properly. This stops tumour cells reproducing. So let's have a talk about how it works then. So a nitrogen atom on a guanine base in DNA forms a coordinating bond. So let's have our guanine here. It forms a coordinated bond with cisplatin's platinum ion, replacing one of the chlorine ligands, and this is a ligand substitution. So we're going to go through a ligand substitution. If you're not sure fully about ligand substitution, we will do a video at some point about transition metals. So when we do a session about transition metals, I will cover it then, and I'll come back to it then and mention that this is what that is. And then what happens is a second nitrogen atom from a nearby guanine, and that's either on the same strand of DNA or the opposite strand, can bond to the platinum and replace the second chloride ion. So the same thing basically happens again. Second nitrogen from another nearby guanine comes and replaces that second chloride ion. The presence of the cisplatin complex bound to the DNA strands causes the strands to kink. This means that the DNA strands can't unwind and then they can't be copied properly. So the cell can't replicate. And this damage to the DNA also triggers mechanisms that leads to cell death. The issue with this, we think, oh great, we can just use this as a cancer drug, but cisplatin can bind to DNA in normal cells as well as cancer cells. So this is a particular problem for any healthy cells replicate frequently. So things such as hair cells and blood cells, because cisplatin stops them from replicating in the same way as it does the cancer cells. And this means that cisplatin can cause hair loss, and it can suppress the immune system, which is controlled by the white blood cells, and it can also lead to kidney damage. These side effects can be lessened by giving a patient very low doses of cisplatin. And another way to reduce the side effects of cisplatin is to target it to the tumour. This means using a method that delivers the drug only to the cancer cells, so it doesn't get the chance to attack healthy cells. Despite the side effects of cisplatin, it is still used as a chemotherapy drug. And this is because the balance of long-term positive effects curing cancer outweigh the negative short-term effects. What we're going to have a look at now is a couple of exam questions. So here we're given the structures of a phosphate group, the base guanine, and the sugar 2-deoxyribose. So you're asked to use the structures to draw a diagram showing a DNA nucleotide containing the base guanine. So we would use our phosphate group first. I haven't actually given myself a lot of space, so this might be a little bit of a nightmare. You can just use it as structural or displayed formula, it doesn't matter. I've got a little OH group in here. Then there's my nitrogen. And what's really important is we know which nitrogen is going to bond on our guanine, which obviously is going to be this one here. There we go. And then you're asked the complementary base of guanine is cytosine. Draw a diagram to show how guanine bonds to cytosine in a DNA double helix and what type of bonds are formed between the bases. So I haven't actually left myself really enough room, but let's try it. And we're going to just draw out guanine first. I'm going to draw out guanine down here. Oops. There we go. Just going to actually, let me write on here. No, okay, Okay, so I'm just going to go through that last bit on a blank slide. So what I'm going to do is draw that cytosine and guanine, join them together. So I'm going to start with cytosine because it's easier up at the top. So you obviously be given this in your data booklet. I would just draw the molecule of cytosine out first of all. I'm awful at drawing these if you hadn't worked that part out by now. There we go. So my NH group here, that's my cytosine drawn out, and that's supposed to be my double bonds. Sort of there. Make sure you've added your lone pairs on, and obviously that oxygen's a lone pair, so we've got negative charge, and add your charges, nitrogen's got lone pair. Add negative charge. That's NH2, and our hydrogen is obviously going to have a positive charge, so add that on there. Let's draw the guanine now. I'm just going to draw the lines of where it's going to join and bond here. It's easy because I can line them up. So that negative lone pair oxygen is going to join with a positive hydrogen off of the guanine. So let's draw that on now. And that will go with a nitrogen and another hydrogen, which is then going to bond with this lone pair, bond with another positive hydrogen here. A lone pair oxygen here with negative charge. Then we just need to finish off the molecule of guanine. Oops. So you've got guanine here, cytosine up here, and then the question asked for all the types of bonds. So the bonds between the bases are hydrogen bonds. So that is an overview of amino acids, proteins, enzymes, and DNA. We will have a session where we just look at some exam questions for these topics. Thank you for watching.